Hello, everyone. Welcome to the UC Botanical Garden at Berkeley's virtual classroom. We're so thrilled to have you all here today. We are going to take a deep dive into the world of newts. So a little bit of information about today's talk. We're going to be on for about an hour, maybe about 50 minutes with some time at the end for questions. My name is Mary. If you have questions throughout this talk, please feel free to pop them in the chat and I will field them over to our speaker towards the end. If you're having any audio or visual trouble, also feel free to pop that in the chat and we'll do our best to support you with that. We do plan to record today's talk, so if you can't join us for the whole thing, feel free to hang on over the next few days and you'll receive a link to be able to view this recording. But without further ado, I would love to open this up to our presenter. Our presenter today is Kannon Pearson, who is a fourth-year PhD student in the Museum of Vertebrate Zoology right here at UC Berkeley. She studies newt toxins, development, and hybridization. When not splashing about in ponds, she can be found drawing amphibians and participating in efforts to diversify and share science. So we're thrilled to welcome you here today, Kannon. Please feel free to take it away, and thank you all for joining us. Oh, special little note, I do have a little 20% off membership, a little gift from our membership team for our February Newt Palooza. So I'll be popping that in the chat, so you'll have that code to get a discount on your membership here at the garden. So without further ado, welcome Kannon to our stage, and I will disappear into the background.
All right. Hi, everybody. I'm going to share my screen really quick. There we go. All right. Yeah, I really appreciate the invite, and I'm so excited to talk to you all about newts today as part of the Botanical Gardens Newt Palooza. The title of my talk kind of alternated between Know Your Newt and this title, Pacific Newts, Poisonous and Punctual. So I suppose choose whichever title you prefer to believe this talk is called. All right, a little bit about me. I'm Kannon Pearson. I'm a fourth-year PhD student in the Museum of Vertebrate Zoology at UC Berkeley. So I'm hosted in the middle of the Valley Life Sciences building in the middle of campus. I'm a herpetologist. I specialize in newts and toads. I'm a PhD student. I'm an artist. I like to mostly draw newts and toads. I really have a fairly narrow range of interests here. And I'm also passionate about advocating for queer inclusion in herpetology and about bringing folks into nature. And here on the right is a picture of me doing some of my standard work activities. In this case, I was in a pond in Idaho trying to trap some newts. There is one pond in, well, two ponds in the entire state of Idaho that contain newts. And over the summer, I went out to try and catch some, and I successfully caught two. So it was great. Here are the contents of my talk. So today we'll be going through what is a newt. I'll be diving into the mystery of Pacific newt toxins, which is the subject of a lot of my research and a topic that I'm very passionate about. We'll discuss the newts in the Japanese pool. Why are they there? Who are they? What are they doing? And lastly, I'll finish with a discussion of my research plans and the plans that my lab has been sort of putting into place for the newts at the garden. This is my first time giving a talk of this length. So if I end up going a little bit short, I suppose we will just have more time for questions. Okay, part one. What is a newt? And there's something I would like to clarify here, and that is that newts are amphibians. They're not reptiles. They have the basic tetrapod body plan. You know, they've got their arms, they've got their legs, they've got a tail. Nothing about their body plan is terribly exaggerated from that basic form. And as a result, plenty of folks see a newt and they think, it's a soft lizard, which taxonomically, not quite true. So lizards are reptiles and newts are amphibians. so they're in the same class as frogs. A little bit about amphibians. Amphibians spend some portion of their life cycle, for most of them, in the water. They tend to have delicate, sensitive skin that they can breathe through, and they undergo typically metamorphosis. So they metamorphose from one life stage, usually a tadpole, or in the case of a salamander, we call it a larval stage, into their adult stage. Of course, there's exceptions to all of these rules. Biology is always messier than we think it is. But those are some of the basic features of this group. And there's three types of amphibians. Everyone knows frogs. Frogs are just, I think, culturally very widespread. And then there are caecilians, which I like to call the secret amphibian group because they are not widely known. These are limbless amphibians that live in the muck, in the bottom of ponds. They live underground, mostly in equatorial regions. I believe there may be a species that can be found in North America, but they're very hard to find because they live underground. And so as a consequence, very little is known about them, and most folks don't know they exist. But they do. They're pretty special. But what I'll be talking about today are salamanders. And these are amphibians that, again, have that basic tetrapod body plan. They've got their arms, their legs, their tail. Same thing with the slimy skin that they can breathe through. Yeah, so salamanders is our focus today. So to review, the supergroup here is amphibians, and there are around 9,000 species of amphibians known today. Unlike birds or mammals, many new amphibians are discovered every year. There's a lot of research in sort of tropical areas where there's incredible biodiversity, and they just keep discovering new species. So expect this number will go up. Within the group of amphibians, there are roughly 800 salamanders. So most known amphibians are frogs, and only like 10% are salamanders. And within that group of salamanders, we have our group of interest today, which is newts. So newts are a kind of salamander, which is a kind of amphibian. And all newts are salamanders. Not all salamanders are newts. And there are some specific things that make a newt stand out from the general grouping of salamander. So some newt characteristics. Adults live on land and return to the water to breed. They have rough, often bumpy skin, and they're often poisonous. And these are all things I'll be talking about throughout the talk today. I like to say that newts are analogous to toads in a lot of ways. So toads are a subgroup of frogs that have rough, bumpy skin, and they're often poisonous. And on the left here, we have a picture of a California newt that I caught in Santa Clara County. So out of the 127 species of newt in the world, 4 of them live in California. These are the red-bellied newts, which are found mostly in Mendocino and north, the rough-skinned newt, which is found in the Bay Area, all the way up to Alaska. So these newts are quite widespread. The Sierra newt, which is found in the Sierras, big surprise, and the California newt, which ranges from just north of the Bay Area all the way down south to near LA. So if you put all these ranges together, you get basically the entire Pacific coast of North America. And that's why the genus that these newts are in, Pterica, is also called the Pacific newt. They're found all throughout the Pacific. There's one more species of newt in the United States. It's the spotted newt. It's found east. So a lot of folks who grew up on the East Coast or some parts of the Midwest may be familiar with those. But in total, most of the newts in the world are not in North America. Most of them are in Europe and in Asia. And it's thought that newts evolved in Eurasia, and they crossed one of the land bridges into North America sometime in the distant past. Okay, now, hopefully we all have somewhat of an idea of what a newt is. It's an amphibian, it's a salamander. They're often poisonous. And now we're going to dive into a discussion of newt poisons. So here we have a newt doing a very special yoga pose that's called the Unken reflex. Now, if you ever go hiking in the redwoods and you look down, you may notice, the carpeting of redwood forest is often this kind of ruddy brownish red color. And the color of the back of a newt blends in really well. They're actually quite camouflaged. They can be very hard to see against that redwood leaf litter, and just against, dark brown ground surfaces in general. But the belly of a Pacific newt is usually a bright yellow to a bright red color. And that is a warning. So when a newt feels particularly threatened, they will lift up their neck, they will lift up their tail, they will sometimes, if they're really excited, lift up their little palms and show that bright coloration. So they go from being camouflaged to being sort of intimidating. They're trying to communicate to predators, I am poisonous. And they're not just a little bit poisonous. So there's an urban legend. I don't know if this has ever been verified, but the story dates back to the early 1900s. There were 3 hunters in the Oregon Coast Range, and they were found dead at their campsite. They had no injuries. There was no sign of struggle. There was honestly no evidence of any wrongdoing. And there they were, dead in their sleeping rolls. The folks who found these people explored the camp, and they looked inside the coffee pot. And what did they see in the coffee pot? But a rough-skinned newt, Tarica granulosa. Apparently, this newt had climbed into the coffee pot at some point, maybe when they were drawing up the water, maybe they just left it sitting out. It had climbed into the coffee pot, fallen in, gotten boiled into the coffee, and killed all 3 hunters. Now, that just remained a story for a long time, and folks didn't really have a widespread awareness that newts had toxins until the work of Victor Twitty in the early 1900s. So Victor Twitty provided the first scientific evidence of newt toxins. He was a fascinating researcher. He is a professor of biology at Stanford University for most of his career. Before then, he spent some time in Europe and I believe on the East Coast. And he started out as an embryologist. So this was a really big field at the time. Folks interested in how animals organize their bodies when they mature. How does a baby know? How does a baby's body know to grow all the limbs at the right rates? How does it coordinate? Why does each of the parts of our body grow in tandem with each other? These were questions that had totally pretty open questions at the time, and Victor Twitty was really interested in this. And I would say his methods are perhaps not as palatable to the modern audience. So most of Twitty's research was done via grafting experiments. So these were experiments where he would take newt larvae. So if you remember, newt larvae are like the tadpole equivalent of a newt. And actually, I need to back up a bit. These weren't just newt larvae. These were salamanders generally. So his original work was in tiger salamanders and spotted salamanders. So he'd take these baby salamanders and he would transplant body parts between different species. So on this example on the left, he transplanted the hind limbs of a tiger salamander and a spotted salamander. Now, tiger salamanders grow much faster than spotted salamanders in this larval stage. And what he found was that this animal, the resulting Frankenstein's monster of a salamander, had all of its body parts grow at the rate you would expect from the parent species. So we have here a tiger salamander with a large upper body and the tiny legs of a spotted salamander. And on the right, we have a spotted salamander with the small upper body of a spotted salamander and the large legs of a tiger salamander. So he's trying to understand how is size regulated. Now, this is a pretty dramatic photo, and he didn't just work with limbs. He also worked with eyes. Once Twitty got to Stanford, he started to incorporate the local newt species into his research. So, there's newts all over the place. They're actually very common in fresh water and a very accessible study specimen. So he got to Stanford. He started working with newts. And for one of his experiments, he was interested in the growth rate of eyes. And so he excised the eye of a tiger salamander and he swapped it with the eye of a California newt larvae. And what he found shocked him. The tiger salamander became paralyzed. And Twitty wondered, is this a feature? Like, is it the immune system that's upset with the eye for some reason? Like, salamanders tend to do really well with grafting experiments. You couldn't really do this in mammals to the same extent because the mammal immune system would reject the graft, but it works really well in salamanders. So it didn't seem to be an immune system issue. Something else was going on. There was something else about the newt eye that was paralyzing this tiger salamander. And the newt larvae, with that transplanted tiger salamander eye, it did just fine. I mean, the eye is a little big for its body, but it was okay. And it wasn't until roughly 40 years later that the culprit for this paralysis was discovered. It turns out newt skin is loaded with a neurotoxin called tetrodotoxin. This toxin is 1,000 times more poisonous than cyanide. At the time of its discovery, it was the most or one of the most toxic known natural substances. And for those who have any familiarity with chemistry, you may notice that tetrodotoxin has these charges on it. So depending on how acidic or basic the solution it's in, you'll get different charges. But regardless, it's a charged molecule. And charged molecules dissolve in water. They dissolve in coffee. Tetrodotoxin, as a result of these charges, behaves in a very particular way. So it can cross easily through aqueous solutions, water-based solutions, but it's stopped by fats, by lipids. And so tetrodotoxin, if it gets in your body, it doesn't cross the blood-brain barrier. Instead, it paralyzes gradually all of your muscles until it reaches the diaphragm, and you lose the ability to breathe. But because tetrodotoxin doesn't cross the blood-brain barrier, it never reaches the brain, so you're fully conscious as you undergo this paralysis. When folks get poisoned by tetrodotoxin, there's no antidote. All they can do is get you on a respirator and wait for your body to gradually remove the toxin from your system. So it's a pretty potent molecule. And as someone who studies chemical ecology and is really interested in poisons, it's one of my favorites. Now, tetrodotoxin is loaded with mysteries. And one of these mysteries is that it actually shows up everywhere. It shows up in marine environments, it shows up in freshwater environments, it shows up in terrestrial environments. If I showed you a map of all the places where tetrodotoxin has been found, it just about covers most of the places in the world. And these are a few examples of animals that have tetrodotoxin. So on the left here, we have pufferfish. And some of you may be familiar with the Japanese dish, fugu, which is a preparation of pufferfish. And sushi chefs who make fugu have to train for years to carefully slice the fish to remove its ovaries and digestive system, because that's where the toxin is stored. And if you eat fugu, typically the desired effect is a faint tingling or numbness of the lips. and that is the incredibly trace amounts of tetrodotoxin acting on your nerves. So yeah, pufferfish have this toxin, and I would say that's the most famous example of another animal with tetrodotoxin, but it's found in other places. So another popular one is the blue-ringed octopus, and these little tiny guys have tons of tetrodotoxin, and that's why they're so incredibly dangerous. They show up in starfish, or I suppose to be more biologically correct, sea stars. So the toxin's found in sea stars. It's found in flatworms, marine, and terrestrial. It is, of course, found in newts. So here at the bottom is an example of a spotted newt from the eastern parts of the US. It's found in newts just like in other regions as well. So newts in Asia also have this toxin. And it's found in toads. So this picture on the bottom right is a harlequin toad, which are found in South and Central America. And these toads have tetrodotoxin, and they have other more classic toad toxins. So this kind of raises the question, how does a molecule as structurally complicated as tetrodotoxin is end up showing up in so many animals in so many places? In fact, if you just go to the bottom of the ocean or to the bottom of a lake and you take a scoop of that sediment and you run that on a chemical analyzer of some kind, you'll often find tetrodotoxin just present in the environment. So how do you get everywhere? And the answer is bacteria. Bacteria are everywhere. And bacteria are the only organisms that have been shown to be able to produce tetrodotoxin. So the thought process is that bacteria have this synthetic ability, and they've formed relationships with all sorts of different animals across the globe. And those relationships have led to the production of tetrodotoxin in those animals' bodies. So in the case of newts, there was a study in 2021 where they swabbed a bunch of rough-skinned newts, and they grew those bacteria in petri dishes. And they found that a few of the strains living in the skin of newts produced tetrodotoxin. And I'm really interested in the relationship between newts and the bacteria in their skin that make their toxins. And so I'm studying that as part of my PhD research. And on the left here, we have a rather fantastical rendition of how newts may get their toxins. So it turns out that newts lay eggs that are loaded with tetrodotoxin. And then the larvae hatch out, and they kind of lose the toxin as they mature. They don't have that much, and they decrease in toxicity as they approach metamorphosis. And then right around the time of metamorphosis, the newts are at their lowest point of toxicity. And after metamorphosis, they start to accumulate the toxin again. And if you raise a newt larvae in a sterile environment, it doesn't get that switch in production. After they metamorphose, they don't start making the toxin. So it seems like metamorphosis is a really critical time for the establishment of a relationship between the newt and their toxin-producing bacteria. So here I have a little illustration I made for a friend of mine in my lab, and it shows some winged bacteria giving a newt that's approaching metamorphosis some molecules of tetrodotoxin. I will say bacteria don't have wings. They're also not this big. There's a lot wrong with this image, but it was fun to make. Okay, so this gets us to another question, which is why are the newts so toxic? I gave you a bunch of examples of different animals that have this toxin, and the newts in California and along the Pacific coast consistently are one of the most toxic animals that have tetrodotoxin. So why? Why do they need that much tetrodotoxin? Well, the answer may be garter snakes. So most predators can't eat newts. If an owl eats a newt, most of the time it dies. And there's folks in the museum that prep owl specimens, and they found newts inside of the carcasses of freshly dead owls that tried to swallow it, and passed away. Invasive bullfrogs, when they try to eat newts, they typically die too. Animals native to the environment, by and large, are not resistant to tetrodotoxin, but some garter snakes are. So these garter snakes have modifications in their body that allow them to resist this toxin. Tetrodotoxin binds to a specific set of voltage-gated sodium channels, which is a lot of scientific words to say that toxin blocks the ability of nerves to send signals. And these snakes have mutations in those sodium channels, such that the toxin, when it approaches the channel, it just bounces off. It doesn't bind. So they've got this predator that can successfully eat them in the environment. And the system of garter snakes and newts is often used as an example of arms race coevolution, which I think is a very sort of exciting framework for evolution. So imagine you're a newt. Well, let's say, imagine you're a population of newts in a specific spot, and you're toxic and no one can eat you. And then the garter snakes around you evolve resistance to your toxin. And now the garter snakes are eating you. So that evolutionary pressure from the garter snakes may result in the less toxic newts getting eaten. So the next generation of newts is going to be more toxic than the previous generation of newts. So now a garter snake tries to eat these extra toxic newts, and it dies. It can't handle it. It's too toxic. What do you do then? Well, all the garter snakes with less toxin resistance die off. And this next generation of garter snakes is more resistant to the toxin. And this is something that's called arms race coevolution, where the prey becomes harder to predate and the predator gets better at eating them. And they just keep going back and forth. And if you look at a map of the Pacific coast, specifically this map on the left, this shows newt toxicity across the coast. You can see they're extra toxic near the South Bay, and they're extra toxic up in Oregon. And if you look on the right, we have a map of garter snake toxin resistance across the coast. And you can see that the areas where the garter snakes are more toxic, these more orangey colors, are also the areas where the newts tend to be more toxic. I think I just called the garter snakes toxic. I didn't mean that. I mean the areas where the garter snakes are more resistant to the toxin are the same areas where the newts have more toxin. And so this is a very cool pattern, and it's the textbook case for this kind of coevolution. And you know, Oregon is right where the coffee pot incident happened. The newts in that area would be toxic enough to kill 3 hunters. In fact, there's newts that have been found that have, I believe, up to 12 milligrams of tetrodotoxin in their entire bodies, which is not enough to just kill 3 adult humans. That's enough to kill 20 adult humans. So this arms race coevolution seems to have created, in some case, just extraordinarily, unbelievably poisonous animals. And I don't know, I think it's pretty cool. All right, now some disclaimers. I know I've been telling you a lot of rather lurid stories about tetrodotoxin and about newts. But you know, if you go out into the wild, you don't need to be scared of newts. They're genuinely only poisonous to you if they are eaten. So you may remember I said the toxin dissolves in water. Well, your skin, the surface of your skin is mostly fats. It can't cross the surface of your skin in large amounts. And you have to really swallow a newt to have any risk of toxicity, or you know, have it crawl into your coffee pot. I will say there's been cases where I've handled maybe 100 or 200 newts in a day. And way back in the beginning of my program, before I knew any better, I used to handle them just with clean, bare hands. And I did notice that at the end of the day, my fingertips did get numb. So, you know, in high enough amounts, it can affect you a little bit through the skin. But by and large, they're safe to handle with clean hands or with gloves. Honestly, the risk of handling newts is not them harming you, it's you harming them. So the skin of newts is fairly sensitive. They breathe through their skin. It's the site where they kind of interact with the environment, and they are at risk of infections in their skin. And the oils and salts and sunscreen on your hands can really irritate the sensitive skin of amphibians in general. So if you're going out in nature, best practice is just look, don't touch. And if you're in a situation where you need to touch the animal, make sure you've washed your hands. There's no moisturizer, no sunscreen, none of that. Or even better, have a pair of gloves, just so that these cute little guys don't suffer from your interaction with them. Okay, so I've told you loads of stories about newt toxins. But now I want to kind of hone in on the place that we all know and love, which is the UC Botanical Gardens, specifically the newts that live in the Japanese pool. If you visit the Japanese pool, which I would assume everyone here has, but if you haven't, you really should. It's beautiful. It's covered in vegetation. There's plants all around it. And if you look into the water, it is clear enough for you to see newts swimming about. And if you visit in the spring, that's when the newts are most abundant. That's when you can see tons of them, often, mating, laying eggs, doing all sorts of exciting things. But I've been to the gardens in September as well, and there's still some stragglers that stick around in the water, possibly year-round. Now remember, I said there are 4 species of newts in California. Only 2 of these species are found in the Japanese pool. That is the rough-skinned newt, Pterica granulosa, and the California newt, Pterica turosa. I'll note really quick that granulosa is, I believe, Latin for like granular or for bumpy. So if you ever need to remember which Latin name goes for which, granulosa is the rough-skinned because it's got more granular skin. But as I was saying, both of these species are found in the Japanese pool, and they can be found in the pool at the exact same time, which is very cool to me. And they're pretty hard to tell apart as adults. I'm going to give you all a quick little tutorial on ways to distinguish these 2 species of newt. One of these ways is their eggs. Rough-skinned newts lay singular eggs, typically attached to a piece of vegetation, whereas California newts lay their eggs in these big crystalline globules of a bunch of eggs all kind of glued together. Again, they like to attach their egg masses to like a stick or a surface. And I've been a newt biologist for at least 4 years, and I've handled thousands of newts, and I've been to many sites, and never in my entire life have I successfully found a rough-skinned newt egg. They are too small. They hide them too well. But the California newt eggs are often very visible. And if you go to the garden, you'll see tons of these egg masses. This is a photo of one of my collaborators, Professor Gary Bucchiarelli. I drove out with him to Quail Ridge Reserve, and we were looking at various newt ponds up there. And Gary reaches into the pond, and he pulls out this branch, and you can see it's just coated in these clusters. It looks like, I don't know, a giant bunch of grapes made out of quartz or something. And so Turosa just lay so many eggs, and they're often so visible. It's just beautiful. All right, another easy way. Actually, I'd say the first way is not very easy. The only easy way to tell these 2 newt species apart is through their babies. So the larvae are quite distinct. Rough-skinned newt larvae have a fairly uniform brown dorsal color. So they're brown all over. They may be a little yellowish on their bellies as they mature, but they're pretty drab, pretty uniform. Whereas California newt larvae have these 2 black racing stripes down their dorsum. and it makes them quite distinguishable. As they approach metamorphosis, however, these stripes fade, and they once again become a little bit tricky to tell apart. So lastly, how do you tell apart adult rough-skinned and California newts? I put this man shrugging here because I think this accurately captures the sentiments of many herpetologists, which is that even the most skilled among us cannot consistently tell the adults apart. It is just quite difficult. They look very similar, and I would say folks who are convinced they can tell them apart are often wrong when we follow up with genetic evidence to verify the identity of a given animal. But there are a few ways that sometimes hold true, and most of these have to do with the shapes and color of their head. So California newts tend to have lighter coloration on their upper and lower eyelids. Typically, it's a color that matches the orange of their bellies. So that orange kind of goes up and around their eyes. That's a California newt. Additionally, California newts tend to have bulgier eyes. So if you hold the newt up and you look straight down, you can see that the eyes tend to stick out past the perimeter of their heads. So that's another way to tell them apart. Rough-skinned newts have the opposite. So they tend to have dark around their eyes, and their eyes are smaller, and they're in line with the perimeter of their heads. And the similarity of these newts leads to a really interesting question, which is, are they interbreeding? Are they more similar to each other because there's some amount of mixture? And there's a little bit of evidence for this. If you go to places where there's just one of these species, they tend to be more distinct than when you go to places where there's both of them. So throughout the Bay Area, they tend to be especially hard to tell apart. But if you go way up north to Oregon or way down south to LA, they kind of follow the more classic distinct characters that you'd expect. Additionally, these newts can successfully breed in captivity, and they show no signs of hybrid breakdown. So that scientist I talked about earlier, Victor Twitty, bred all the different species of Pacific newt together, and they do just fine. The hybrids do great. There's no sterility, there's no hybrid breakdown, and they can breed back with either parent species. Just as a quick note, once Twitty got out of embryology and he went into breeding newts, he did some truly wild things. One of these things was he actually released tens of thousands of artificial hybridized newts into a stream up in Sonoma County, which, you know, nowadays I think you would classify as almost akin to eco-terrorism in terms of the impact that could have in an environment. So I highly recommend folks look into the research of Twitty. But back to the talk. I'm exploring this question as part of my thesis research. So right now I'm doing field work. I'm going out and about. I'm catching newts. We're scoring their morphological characters. And I am doing genetics to see if they are hybridizing at all in these sympatric areas. All right, so what are they doing in the pond? Well, the answer is hanky-panky. The newts come to the pond to breed. On the right, we have a picture of a male rough-skinned newt holding a female in emplexus. That's their mating grasp. They typically do emplexus before fertilization. So they'll emplex, and if it's successful, then the male will deposit a sperm packet and the female will pick it up. Most adults leave the pond in the summer, though some stick around. And the newts that have left the pond typically sort of disappear. It's thought that they mostly go underground and they hide out and wait for the next rainy season to come out. If you ever see a newt in its sort of dry terrestrial mode, you'll notice it can look really different from an aquatic newt. So as rainy season comes around, the male newts in particular sort of activate their sexy water mode. They become more gelatinous their skin gets smoother, their body gets puffy, their tails get really wide to help them swim more effectively and chase the girlies. And they basically become these like giant slimy gummy bears. It's super, super cute. Now, another cool thing about the newts at the Botanical Garden is that they're the same newts as last year. Effectively, newts have a very strong homing behavior. They always, or almost always, return to the same spot to breed every time. And once again, the person who figured this out was Victor Twitty. So he did all sorts of research into newt homing behavior. And he found that if he took a newt and he flew it out in any direction away from the place where it was breeding, it would, upon being set down, turn around and make a beeline straight back to the breeding site. They can find their breeding site over great distances. So he'd put newts as far away as 8 miles from the streams where he found them, and those newts would come back. He also did some more rather, I would say, difficult experiments where he found that blinded newts can still find their ways back to the pond. Though if a newt had an impaired sense of smell, it did seem to have more difficulty getting back. So smell seems at least somewhat important. I'd say magnetism is another likely explanation for how they navigate so effectively. Though, you know, since the time of Twitty, no one has very thoroughly investigated this question for Pacific newts. All right, so briefly, now that we've talked about what a newt is, we've talked about newt toxins, we've talked about the newts at the Botanical Gardens, I now want to touch upon some of the research that my lab, the Tarvin lab, is planning at the gardens. So it's really cool and very convenient to have a nearby population of one of our study species. It's pretty easy to get to the Botanical Gardens. There's lots of newts there. And the folks at the Botanical Gardens have been very welcoming to our inquiries about doing research projects with them. So we've sort of started developing a lovely collaboration between the Botanical Gardens and the Tarvin lab. And one of our sort of basic initial questions is just, how does the population of the Japanese pool change over time? So one of the ways we're approaching this question is through mark recapture studies using pit tags. So you know how when you adopt a dog or a cat, the vet will offer to microchip that dog or cat. It's the same principle here. We basically microchip the newts, and then we can use this reader, shown on the right, to tell us who the newt is. You know, newts look pretty similar to humans. We can't tell them apart that well, but if you microchip them, you can track the same individual newt across many years, see how often it returns to the pond, see when it stops coming back to the pond. You can get an estimate for how long that newt has lived. You can track features of that newt and how they change over time. So we've been kind of creating ideas for ways to follow up on this research. One of the ideas is monitor the newts for disease. So newts everywhere are declining right now. There's a mysterious newt wasting disease that has shown up in places where newts suddenly become emaciated and they have mass die-offs. So we could track individual newts over the years and see how their disease load changes. We can also track the toxicity of a newt. You know, does one newt become more toxic as the years go by? One of the questions that one of my undergraduate honor students is interested in is studying whether tetrodotoxin from the newts leach into the environment. So maybe, you know, during breeding season, tons of newts show up in the pond. And if we look at the toxin in the water, does that increase during the breeding season? And what implications does that have for the other animals that live there? And we're open to ideas from the public. You know, what do you want to know about the newts at the Botanical Gardens? All of these are just possibilities at this point, and they're just things that we've started discussing. But I think we're really excited to have formed this relationship with the botanical gardens and to see what comes out of that. And if you want to sort of reach out and share some of your ideas, I'll be giving my contact information at the end of this talk. Okay, another quick opportunity. If you're a Berkeley student and you are excited about any of the things I've talked about, I regularly bring Berkeley students as volunteers on my field trips throughout the Bay Area to catch newts and to do this work. So you can reach out to me about that as well, and I'll add you to my mailing list. And again, my contact info is at the end. And here's a picture of me teaching my volunteers how to do the Unken reflex. Unfortunately, humans don't have poisons for the most part, and we don't have brightly colored bodies, but I think it's a quite fun yoga pose to do. And there's other ways to get involved if you're excited about newts. There's programs like the Chileno Valley Newt Brigade, which is where folks go out during the breeding season, and they help the newts cross the road. Newts migrate in huge numbers to get to their breeding ponds, and they often have to cross inhospitable surfaces like roads, and incredible numbers of newts die by getting run over. So there's programs in Chileno Valley and in other places in California where folks gather at night and they just carry them across the road. And I think it's a really beautiful way to help an animal. Additionally, just generally, the Museum of Vertebrate Zoology helps run this website called AmphibiaWeb, which is a database of amphibians. It's like the Wikipedia for amphibians. And we're always looking for more volunteers or for people to donate funds to help keep that afloat. And lastly, as the youths like to say, I highly recommend folks go outside. I think Gen Z tends to call this touching grass. Just go out to the gardens, go see the animals. There is so much more mystery in nature than you could ever conceive of. And every time I go out, I find something new and something beautiful. So if you're already doing that's great. More power to you. And if not, it's really a fun thing to do. All right, that concludes my talk. You can reach me at Kannon Pearson at berkeley.edu. I'm also on Blue Sky, where I post pretty regularly about newts that I found in the field. And I like to post my art there as well. And lastly, if you go to the Tarban Lab website, my bio is listed on there. And if you're interested in sort of topics about herpetology and gay frog memes, I have a talk about that. And there's a YouTube link linked in my bio. So to any queer folks listening, I made that for you. And at this point, I'll be happy to field any questions.
Oh my goodness. Thank you so much, Kannon. I absolutely loved hearing what you had to say. I want to just start off by saying we've received so many questions and so much curiosity on this topic. So thank you so much. You have obviously spurred our imaginations. One of the things I wanted to mention is a lot of folks mentioned that they really appreciated and enjoyed the art that you made on those slides. Truly special, especially the winged creatures. That got a lot of attention. So you kind of touched on this, but one of the things folks were noticing is, yeah, concentration of newts in the water, and could that have an effect on toxicity for drinking the water, touching the water? Definitely got our imaginations going on that, and folks were curious if you had any theories on that.
Yeah, so I do. This is an area that I'm very excited about, and I'm very excited that one of my undergraduate mentees is going to possibly do a research project on it. But if you take a newt and you put it in a container, some amount of toxin does leach out of its body. And newts actually eat larvae, like they eat their own babies. They're cannibals. And the newt larvae can smell tetrodotoxin in the water. And that's how they know that the cannibalistic adults are nearby. So they smell the toxin and they swim away. So we have evidence that they do leach some amount of toxin from their bodies. And the way my undergrad wants to approach this question is to look at toxin levels in the water and see if that increases at all during the breeding season. and also look at toxin levels in the sediment. So of course, like if it's in the water, it's kind of going to get quite diluted over time, but we kind of wonder if maybe it might get locked in the sediment and you might find it in higher concentrations in there. I would say again, there's basically no risk to humans from this. It's a very, very small amount of toxin, but we think it might be ecologically relevant for like maybe animals that live in the mud, and that would be so exciting.
Fascinating. On the theme of toxins, some of us were wondering, so if some garter snakes are able to process this toxin or at least consume it, is there any possibility that they're retaining any of this toxicity themselves?
So that is a lovely question. And actually, I'm going to scroll manically all the way to my garter snake picture. You look at a garter snake, a lot of the species of garter snakes have pretty potent coloration that sort of reminds you of the colors you might find in a poisonous animal. They've got like these red spots. I highly recommend everyone Google the San Francisco garter snake. It's the most beautiful snake in the world. And those are bright blue and red. They also eat newts. And it's been found that the garter snake liver does accumulate some amount of toxin from the newts. It's not known if this is enough to like affect their predators. And it's not known if this is like an evolution, an adaptation, or if this is just an accidental byproduct. If you eat anything, you know, the chemicals from that pass through your liver at some point or get stored in your liver. But they do accumulate some amount of toxin. And there's been work done by Chris Feldman in this area, if anyone's interested in learning more.
Interesting. And then some folks were curious, you were talking a bit about the wasting disease. Do you have any theories or is there anything percolating in the science field about what this could be? Or is it too early in the stages to really get into theories?
Yeah, so I'm not as familiar with the current research on this, but there's a lab at San Francisco State led by Vance Friedenberg that's kind of at the cutting edge. Some of the theories I've seen is that it's a result of chytridiomycosis, which is a disease that has affected amphibians worldwide. It's the amphibian pandemic. I'm not sure if there's evidence of that at this point, but other theories is just like more toxins in the water, or maybe climate change affects the availability of prey items. So newts mostly eat small bugs, and climate change and just human impacts have actually devastatingly wiped out huge amounts of small insects. And that's not something we notice as easily, but there's like way less, and they may show up at the wrong times. So if they show up at the wrong times, the newts aren't going to get the food they need when they're in their breeding condition. So there's been speculation, but we don't know yet what exactly is happening.
And on the other side of that, we've had a few folks say they can tell you're passionate about it and they're so curious about it. Why newts? What got you into this field? Where'd that start?
Yeah. So again, going way back to my tetrodotoxin slide. Here I cite a paper, Vialli et al. 2021, and this paper did it. I read that paper as an undergraduate, and that's the paper where they cultivated the bacteria from the skin of newts, and they found they made the toxin. And I had been really interested in chemistry as an undergrad, and also in molecular biology. And then I took a herpetology class with Jim McGuire at the museum, and I got really excited about amphibians. So I knew I liked chemicals. I liked how organisms interact with each other. I like how molecules work, and I love amphibians. And this paper is just like the perfect combination of all those things, where just the idea of a toxin sort of mediating so many relationships between animals and also being the result of a relationship between bacteria and the newt was super exciting to me.
Well, we share your passion. I want to let folks know while we're on the topic of newts that over the next 2 Sundays for our Newtapalooza celebration, we actually have a newt discovery station where you can go get an up-close look with one of our docents at what is happening in this pond. There will be some folks there between the hours of 12 and 3 at the pond. We also have an accessible station that will be at the tour deck if folks want to visit that one. We'll have both available from 12 to 3 or the next 2 Sundays. But I want to say thank you so much, Kannon. This was so much fun. I learned so much. I can't wait to share some of this information with our summer campers and just every general visitor who walks through our gate. It's a great time to be at the garden. We love the rain. The rain and the newts are such a happy connection. So while it does change the aesthetic, it sure does bring the newts to the pond. So again, thank you so much. And I hope you all enjoy the rest of this week. I'll be sending a recording out if folks missed any section of that. Please feel free to enjoy the recording. I'll also send you that 20% off coupon for membership, and I'll include the dates for the Newt Palooza Discovery Stations that'll be at the garden.
Yeah.
And I also, sorry, last thing I want to mention is, Kannon, there have been so many comments in the chat that I'll share with you as well. Some really wonderful things that folks are sharing. So I will make sure that makes it to you as well.
Yeah, and thank you everyone for showing up and being curious about newts. Again, you can always reach out to me at my school email. I've just now seeing that some folks sent me some questions via direct message. And so my apologies that I couldn't get to all of those today. And if you want to inquire further, I will happily answer over email. And I just really love that folks love the newts, and I really appreciate you all for showing up.
Thank you. Have a wonderful day, everyone. Thanks, Kannon.